Examples of enzymatic reactions. Thus far we have focused on the general principles of catalysis, and on introducing some of the kinetic parameters used to describe enzyme action. We now turn to several examples of specific enzyme reaction mechanisms. To understand the complete mechanism of action of a purified enzyme, we need to identify all substrates, cofactors, products, and regulators. We also need to know 1. The temporal sequence in which enzyme-bound reaction intermediates form. 2. The structure of each intermediate and each transition state. 3. The rates of interconversion between intermediates. 4. The structural relationship of the enzyme to each intermediate. And 5. The energy contributed by all reacting and interacting groups to the intermediate complexes and transition states. There are still few enzymes for which we have an understanding that meets all these requirements. We present here the mechanisms for 4 enzymes: chymotrypsin, hexokinase, enolase, and lysozyme. These examples are not intended to cover all possible classes of enzyme chemistry. They are chosen in part because they are among the best understood enzymes and in part because they clearly illustrate some general principles outlined in this chapter. The discussion concentrates on selected principles, along with some key experiments that have helped to bring these principles into focus. We use the chymotrypsin example to review some of the conventions used to depict enzyme mechanisms. Much mechanistic detail and experimental evidence is necessarily omitted. No one book could completely document the rich experimental history of these enzymes. In addition, we consider only briefly the special contribution of coenzymes to the catalytic activity of many enzymes. The function of coenzymes is chemically varied, and we describe each coenzyme in detail as it is encountered in Part 2. The chymotrypsin mechanism involves acylation and deacylation of a SER residue. Bovine pancreatic chymotrypsin, Mr. 25191, is a protease, an enzyme that catalyzes the hydrolytic cleavage of peptide bonds. This protease is specific for peptide bonds adjacent to aromatic amino acid residues, TRP-Phe-TIR. The three-dimensional structure of chymotrypsin is shown in Figure 6-20, with functional groups in the active site emphasized. The reaction catalyzed by this enzyme illustrates the principle of transition state stabilization and also provides a classic example of general acid-base catalysis and covalent catalysis. Chymotrypsin enhances the rate of peptide bond hydrolysis by a factor of at least 10⁹. It does not catalyze a direct attack of water on the peptide bond. Instead, a transient covalent acylenzyme intermediate is formed. The reaction thus has 2 distinct phases. In the acylation phase, the peptide bond is cleaved and an ester linkage is formed between the peptide carbonyl carbon and the enzyme. In the deacylation phase, the ester linkage is hydrolyzed and the non-acylated enzyme regenerated. Figure 6-20's structure. The first evidence for a covalent acylenzyme intermediate came from a classic application of pre-steady-state kinetics. In addition to its action on polypeptides, chymotrypsin also catalyzes the hydrolysis of small esters and amides. These reactions are much slower than hydrolysis of peptides, because less binding energy is available with smaller substrates. The pre-steady state is also correspondingly longer, thus simplifying the analysis of the resulting reactions. Investigations by B.S. Hartley and B.A. Kilby in 1954 found that chymotrypsin hydrolysis of the ester p-nitrophenylacetate, as measured by release of p-nitrophenol, proceeds with a rapid burst before leveling off to a slower rate, Fig. 6-21. By extrapolating back to zero time, they concluded that the burst phase corresponded to the release of just under 1 molecule of p-nitrophenol for every enzyme molecule present. A small fraction of their enzyme molecules were inactive. Hartley and Kilby suggested that this release of p-nitrophenol occurred during a rapid acylation of all the enzyme molecules, with the rate for subsequent turnover of the enzyme limited by a subsequent, slower deacylation step. Similar results have since been obtained with many other enzymes. The observation of a burst phase provides yet ANOTHEREXAMPLEOFTHEUSEOFKINETICSTOBREAKDOWNA CO and S. Figure 6 to 21. Additional features of the chymotrypsin mechanism have been discovered by analyzing the dependence of the reaction on pH. The rate of chymotrypsin catalyzed cleavage generally exhibits a bell-shaped pH rate profile, Fig. 6 to 22. The rates plotted in Figure 6-22 are obtained at low, subsaturating, substrate concentrations and therefore represent Cat/Km, CX 6 to 27, p. 203. A more complete analysis of the rates at different substrate concentrations at each pH allows researchers to determine the individual contributions of the Cat and Km terms. After obtaining the maximum rates at each pH, one can plot the Cat alone versus pH, Fig. 6-22b. After obtaining the Km at each pH, researchers can then plot 1/Km versus pH, Fig. 6-22c. Kinetic and structural analyses have revealed that the change in Cat reflects the ionization state of His 57. The decline in Cat at low pH results from protonation of His 57, so that it can no longer extract a proton from Ser 195 in step 2 of the reaction, see Fig. 6 to 23. This rate reduction illustrates the importance of general acid and general base catalysis in the mechanism for chymotrypsin. The changes in the 1/Km term reflect the ionization of the alpha amino group of Al16 at the amino terminal end of one of the enzyme's 3 polypeptide chains. This group forms a salt bridge to ASP194, stabilizing the active conformation of the enzyme. When this group loses its proton at high pH, the salt bridge is eliminated, and a conformational change closes the hydrophobic pocket where the aromatic amino acid side chain of the substrate inserts, Fig. 6 to 20. Substrates can no longer bind properly, which is measured kinetically as an increase in Km. Figure 6 to 22. Alpha amino group. As shown in Figure 6 to 23, the nucleophile in the acylation phase is the oxygen of Ser 195. Proteases with a Ser residue that plays this role in reaction mechanisms are called serine proteases. The pKa of a Ser hydroxyl group is generally too high for the unprotonated form to be present in significant concentrations at physiological pH. However, in chymotrypsin, Ser 195 is linked to as 57 and ASP102 in a hydrogen bonding network referred to as the catalytic triad. When a peptide substrate binds to chymotrypsin, a subtle change in conformation compresses the hydrogen bond between His 57 and ASP102, resulting in a stronger interaction, called a low-barrier hydrogen bond. This enhanced interaction increases the pKa of His 57 from 7, for free histidine, to 0.12, allowing the His residue to act as an enhanced general base that can remove the proton from the Ser 195 hydroxyl group. Deprotonation prevents development of a highly unstable positive charge on the Ser 195 hydroxyl and makes the Ser side chain a stronger nucleophile. At later reaction stages, His 57 also acts as a proton donor, protonating the amino group in the displaced portion of the substrate, the leaving group. As the Ser 195 oxygen attacks the carbonyl group of the substrate, Fig. 623, step 2, a very short-lived tetrahedral intermediate is formed in which the carbonyl oxygen acquires a negative charge. This charge, forming within a pocket on the enzyme called the oxyanion hole, is stabilized by hydrogen bonds contributed by the amide groups of 2 peptide bonds in the chymotrypsin backbone. One of these hydrogen bonds, contributed by GLY193, is present only in this intermediate and in the transition states for its formation and breakdown. It reduces the energy required to reach these states. This is an example of the use of binding energy in catalysis through enzyme transition state complementarity. An understanding of protease mechanisms leads to new treatments for HIV infections. New pharmaceutical agents are almost always designed to inhibit an enzyme. The extremely successful therapies developed to treat HIV infections provide a case in point. The human immunodeficiency virus, HIV, is the agent that causes acquired immune deficiency syndrome, AIDS. In 2015, an estimated 34 to 41 million people worldwide were living with HIV infections, with about 2 million new infections that year and approximately 1.2 million fatalities. AIDS first surfaced as a worldwide epidemic in the 1980s. HIV was discovered soon after and was identified as a retrovirus. Retroviruses possess an RNA genome and an enzyme, reverse transcriptase, capable of using RNA to direct the synthesis of a complementary DNA. Efforts to understand HIV and develop therapies for HIV infection benefited from decades of basic research, both on enzyme mechanisms and on the properties of other retroviruses. Mechanism Figure 6-23 Hydrolytic cleavage of a peptide bond by chymotrypsin. 4. Formation. Figure 6-24 Mechanism of action of HIV protease. A retrovirus such as HIV has a relatively simple life cycle, see Fig. 2632. Its RNA genome is converted to duplex DNA in several steps catalyzed by the reverse transcriptase, described in Chapter 26. The duplex DNA is then inserted into a chromosome in the nucleus of the host cell by the enzyme integrase, described in Chapter 25. The integrated copy of the viral genome can remain dormant indefinitely. Alternatively, it can be transcribed back into RNA, which can then be translated into proteins to construct new virus particles. Most of the viral genes are translated into large polyproteins, which are cut by an HIV protease into the individual proteins needed to make the virus, see Fig. 26-33. Only 3 key enzymes operate in this cycle: the reverse transcriptase, the integrase, and the protease. These enzymes thus represent the most promising drug targets. There are 4 major subclasses of proteases. The serine proteases, such as chymotrypsin and trypsin, and the cysteine proteases, in which a cis residue serves a catalytic role similar to that of ser in the active site, form covalent enzyme substrate complexes, the aspartyl proteases and metalloproteases do not. The HIV protease is an aspartyl protease. 2 active site ASP residues facilitate the direct attack of a water molecule on the carbonyl group of the peptide bond to be cleaved, Fig. 6-24. The initial product of this attack is an unstable tetrahedral intermediate, much like that in the chymotrypsin reaction. This intermediate is close in structure and energy to the reaction transition state. The drugs that have been developed as HIV protease inhibitors form non-covalent complexes with the enzyme, but they bind to it so tightly that they can be considered irreversible inhibitors. The tight binding is derived in part from their design as transition state analogues. The success of these drugs makes a point worth emphasizing. The catalytic principles we have studied in this chapter are not simply abstruse ideas to be memorized, their application saves lives. The HIV protease is most efficient at cleaving peptide bonds between Phe and pro residues. The active site has a pocket that binds an aromatic group next to the bond to be cleaved. Several HIV protease inhibitors are shown in Figure 6-25. Although the structures appear varied, they all share a core structure, a main chain with a hydroxyl group positioned next to a branch containing a benzyl group. This arrangement targets the benzyl group to an aromatic, hydrophobic, binding pocket. The adjacent hydroxyl group mimics the negatively charged oxygen in the tetrahedral intermediate in the normal reaction, providing a transition state analog. The remainder of each inhibitor structure was designed to fit into and bind to various crevices along the surface of the enzyme, enhancing overall binding. The availability of these effective drugs has vastly increased the lifespan and quality of life of millions of people with HIV and AIDS. In early 2015, 15 million of the approximately 37 million people living with HIV infection were receiving antiretroviral therapy. Hexokinase undergoes induced fit on substrate binding yeast. Hexokinase, Mr. 107862, is a bisubstrate enzyme that catalyzes the reversible reaction. Figure 6. ATP and ADP always bind to enzymes as a complex with the metal ion Mg2+. In the hexokinase reaction, the gamma phosphoryl of ATP is transferred to the hydroxyl at C6 of glucose. This hydroxyl is similar in chemical reactivity to water, and water freely enters the enzyme active site. Yet hexokinase favors the reaction with glucose by a factor of 10⁶. The enzyme can discriminate between glucose and water because of a conformational change in the enzyme when the correct substrate binds, Fig. 6 to 26. Hexokinase thus provides a good example of induced fit. When glucose is not present, the enzyme is in an inactive conformation, with the active site amino acid side chains out of position for reaction. When glucose, but not water, and Mg-ATP bind, the binding energy derived from this interaction induces a conformational change in hexokinase to the catalytically active form. Figure 6 to structure. Induced by Anderson. This model has been reinforced by kinetic studies. The 5-carbon sugar xylose, stereochemically similar to glucose, but 1 carbon shorter, binds to hexokinase, but in a position where it cannot be phosphorylated. Nevertheless, addition of xylose to the reaction mixture increases the rate of ATP hydrolysis. Evidently, the binding of xylose is sufficient to induce a change in hexokinase to its active conformation, and the enzyme is thereby tricked into phosphorylating water. The hexokinase reaction also illustrates that enzyme specificity is not always a simple matter of binding one compound but not another. In the case of hexokinase, specificity is observed not in the formation of the ES complex, but in the relative rates of subsequent catalytic steps. Reaction rates increase greatly in the presence of a substrate, glucose, that is able to accept a phosphoryl group. Induced fit is only one aspect of the catalytic mechanism of hexokinase. Like chymotrypsin, hexokinase uses several catalytic strategies. For example, the active site amino acid residues, those brought into position by the conformational change that follows substrate binding, participate in general acid-base catalysis and transition state stabilization. The enolase reaction mechanism requires metal ions. Another glycolytic enzyme, enolase, catalyzes the reversible dehydration of 2-phosphoglycerate to phosphoenol pyruvate. The reaction provides an example of the use of an enzymatic cofactor, in this case a metal ion. An example of coenzyme function is provided in box 6 to 3. Yeast enolase, Mr. 93316, is a dimer with 436 amino acid residues per subunit. The enolase reaction illustrates one type of metal ion catalysis, and provides an additional example of general acid-base catalysis and transition state stabilization. The reaction occurs in 2 steps, Fig. 6-27a. First, List 345 acts as a general base catalyst, abstracting a proton from C2 of 2-phosphoglycerate, then Glu 211 acts as a general acid catalyst, donating a proton to the OH leaving group. The proton at C2 of 2-phosphoglycerate is not acidic, and thus is quite resistant to its removal by List 345. However, the electronegative oxygen atoms of the adjacent carboxyl group pull electrons away from C2, making the attached protons somewhat more labile. In the active site, the carboxyl group of 2-phosphoglycerate undergoes strong ionic interactions with 2 bound Mg2+ ions, Fig. 6-27b, greatly enhancing the electron withdrawal by the carboxyl. Together, these effects render the C2 proton sufficiently acidic, lowering the pKa, that one proton can be abstracted to initiate the reaction. As the unstable enolate intermediate is formed, the metal ions further act to shield the 2 negative charges on the carboxyl oxygen atoms that transiently exist in close proximity to each other. Hydrogen bonding to other active site amino acid residues also contributes to the overall mechanism. The various interactions effectively stabilize both the enolate intermediate and the transition state preceding its formation. Mechanism Figure 6 to 27. 2-step lysozyme uses 2 successive nucleophilic displacement reactions. Lysozyme is a natural antibacterial agent found in tears and egg whites. The hen egg white lysozyme, Mr. 14296, is a monomer with 129 amino acid residues. This was the first enzyme to have its three-dimensional structure determined by David Phillips and colleagues in 1965. The structure revealed 4 stabilizing disulfide bonds and a cleft containing the active site, Fig. 628A. More than 5 decades of investigations have provided a detailed picture of the structure and activity of the enzyme, and an interesting story of how biochemical science progresses. The substrate of lysozyme is peptidoglycan, a carbohydrate found in many bacterial cell walls. Lysozyme cleaves the beta-1,4 glycosidic CO bond, P258, between the two types of sugar residues in the molecule N-acetylmuramic acid, MOR2AC, and N-acetylglucosamine, GLCNAC, Fig. 6. 28b, often referred to as NAM and NAG, respectively, in the research literature on enzymology. 6 residues of the alternating MOR2AC and GLCNAC in peptidoglycan bind in the active site, in binding sites designated A through F. Model building has shown that the lactyl side chain of MOR2AC cannot be accommodated in sites C and E, restricting MOR2AC binding to sites B, D, and F. Only one of the bound glycosidic bonds is cleaved, that between a MOR2AC residue in site D and a GLCNAC residue in site E. The key catalytic amino acid residues in the active site are GLU35 and ASP52, Fig. 629A. The reaction is a nucleophilic substitution, with OH from water replacing the GLCNAC at C1 of MOR2AC. Fig. Shown, with the lysozyme mechanism Figure 6 to the mechanism, with the active site residues identified and a detailed structure of the enzyme available, the path to understanding the reaction mechanism seemed open in the 1960s. However, definitive evidence for a particular mechanism eluded investigators for nearly 4 decades. There are 2 chemically reasonable mechanisms that could generate the product observed when lysozyme cleaves the glycosidic bond. Phillips and colleagues proposed a dissociative, SN1 type, mechanism, Fig. 6-29A, left, in which the GLCNAC initially dissociates in step 1 to leave behind a glycosyl cation, a carbocation intermediate. In this mechanism, the departing GLCNAC is protonated by general acid catalysis by GLU35, located in a hydrophobic pocket that gives its carboxyl group an unusually high pKa. The carbocation is stabilized by resonance involving the adjacent ring oxygen, as well as by electrostatic interaction with the negative charge on the nearby ASP52. In step 2, water attacks at C1 of MOR2AC to yield the product. The alternative mechanism, Fig. 6-29, GLCNAC. As in the first mechanism, GLU35 acts as a general acid to protonate the departing GLCNAC. In step 2, water attacks at C1 of MOR2AC to displace the ASP52 and generate product. The Phillips mechanism, SN1, was widely accepted for more than 3 decades. However, some controversy persisted and tests continued. The scientific method sometimes advances an issue slowly, and a truly insightful experiment can be difficult to design. Some early arguments against the Phillips mechanism were suggestive, but not completely persuasive. For example, the half-life of the proposed glycosyl cation was estimated to be 10-12 seconds, just longer than a molecular vibration, and not long enough for the needed diffusion of other molecules. More important, lysozyme is a member of a family of enzymes called retaining glycosidases, all of which catalyze reactions in which the product has the same anomeric configuration. As the substrate, anomeric configurations of carbohydrates are examined in Chapter 7, and all of which are known to have reactive covalent intermediates like that envisioned in the alternative, SN2, pathway. Hence, the Phillips mechanism ran counter to experimental findings for closely related enzymes. A compelling experiment tipped the scales decidedly in favor of the SN2 pathway, as reported by Stephen Withers and colleagues in 2001. Making use of a mutant enzyme, with residue 35 changed from GLU to GLN and artificial substrates which combined to slow the rate of key steps in the reaction, these workers were able to stabilize the elusive covalent intermediate. This allowed them to observe the intermediate directly, using both mass spectrometry and X-ray crystallography Fig. 6-29b. Is the lysozyme mechanism now proven? No. A key feature of the scientific method, as Albert Einstein once summarized it, is no amount of experimentation can ever prove me right, a single experiment can prove me wrong. In the case of the lysozyme mechanism, one might argue, and some have, that the artificial substrates, with fluorine substitutions at C1 and C2, as were used to stabilize the covalent intermediate, might have altered the reaction pathway. The highly electronegative fluorine could destabilize an already electron-deficient oxocarbenium ion in the glycosyl cation intermediate that might occur in an SN1 pathway. However, the SN2 pathway is now the mechanism most in concert with available data. An understanding of enzyme mechanism produces useful antibiotics. Penicillin was discovered in 1928 by Alexander Fleming, but it was another 15 years before this relatively unstable compound was understood well enough to use it as a pharmaceutical agent to treat bacterial infections. Penicillin interferes with the synthesis of peptidoglycan, the major component of the rigid cell wall that protects bacteria from osmotic lysis. Peptidoglycan consists of polysaccharides and peptides cross-linked in several steps that include a transpeptidase reaction, Fig. 6-30. It is this reaction that is inhibited by penicillin and related compounds, Fig. 631a, all of which are irreversible inhibitors of transpeptidase, able to bind its active site through a segment that mimics one conformation of the diala segment of the peptidoglycan precursor. The peptide bond in the precursor is replaced by a highly reactive beta-lactam ring in the antibiotic. When penicillin binds to the transpeptidase, an active site SER attacks the carbonyl of the beta-lactam ring and generates a covalent adduct between penicillin and the enzyme. The leaving group remains attached, however, because it is linked by the remnant of the beta-lactam ring, Fig. 631b. The covalent complex irreversibly inactivates the enzyme. This, in turn, blocks synthesis of the bacterial cell wall, and most bacteria die as the fragile inner membrane bursts under osmotic pressure. Human use of penicillin and its derivatives has led to the evolution of strains of pathogenic bacteria that express beta-lactamases, Fig. 632a, enzymes that cleave beta-lactam antibiotics, rendering them inactive. The bacteria thereby become resistant to the antibiotics. The genes for these enzymes have spread rapidly through bacterial populations under the selective pressure imposed by the use, and often overuse, of beta-lactam antibiotics. Human medicine responded with the development of compounds such as clavulanic acid, a suicide inactivator, which irreversibly inactivates the beta-lactamases, Fig. 632b. Clavulanic acid mimics the structure of a beta-lactam antibiotic and forms a covalent adduct with a SER in the beta-lactamase active site. This leads to a rearrangement that creates a much more reactive derivative, which is subsequently attacked by another nucleophile in the active site to irreversibly acylate the enzyme and inactivate it. Amoxicillin and clavulanic acid are combined in a widely used pharmaceutical formulation with the trade name Augmentin. The cycle of chemical warfare between humans and bacteria continues unabated. Strains of disease-causing bacteria that are resistant to both amoxicillin and clavulanic acid have been discovered. Mutations in beta-lactamase within these strains render it unreactive to clavulanic acid. The development of new antibiotics promises to be a growth industry for the foreseeable future. Figure 6-30 The transpeptidase reaction. Figure 6-30. Figure 6-30. Chemical mechanism of beta-lactam. Summary 6.4 Examples of enzymatic reactions. Chymotrypsin is a serine protease with a well-understood mechanism, featuring general acid-base catalysis, covalent catalysis, and transition state stabilization. Hexokinase provides an excellent example of induced fit as a means of using substrate binding energy. The enolase reaction proceeds via metal ion catalysis. Lysozyme makes use of covalent catalysis and general acid catalysis, as it promotes two successive nucleophilic displacement reactions. Understanding enzyme mechanism allows the development of drugs to inhibit enzyme action.